Good morning. Thank you for inviting me to present at this amazing conference. I have been listening to a variety of topics today, and I'm so excited that there is so much science going around me. And may I share my slides?
Ma'am, we will first introduce myself, Dr. Lakshmi Jatia.
Okay.
Hello, ma'am. Dr. Chaya, how are you?
I'm good, thank you. Nice to see you.
Welcome all delegates, distinguished speakers, and our chairpersons for this session. Let me introduce the first, our chairperson for today, Dr. Janit Berlin-Raj. I'm very proud to say that Dr. Janit Berlin-Raj is alumnus of our college, Arpade Vidu Medical College. Welcome, Dr. Janit. And he is now, we are very proudly to say that he is heading the Department of Physiology in another medical college. And apart from his qualifications in physiology, he has got so many other qualifications that he has in medical education related to research and other clinical subjects. And he is having so many other academic qualifications and responsibilities in his college. He is a member of Board of Studies. He's a member of Board of Examinations, and so many responsibilities he's sharing. And apart from that, we are happy to say that he is also a recipient of Dr. Lalita Kameshwaran Best Research Paper Award. And he is interested in pulmonary functions and diabetes, medical education. That probably is the result of his additional qualifications that he has undergone. And I also see that he's interested in alternative medicine. That is probably the result of his association and his guru, Dr. Madan Mohan Trakru. So we are happy to have you, Dr. Janit, here. Over to you, Dr. Vidya.
Thank you, madam, for that introduction.
Yeah.
Yes.
You have to say something, Dr. Janit?
After
Okay.
Telisir's
Okay. Okay, fine. Okay.
Good morning, everyone. I'm Dr. Vidya Lakshmi, and it's a great honor and privilege for me to introduce Dr. Chaya Gopalan who is a professor and head of Department of Physiology of Manakala Vinayagar Medical College, Medical and Hospital, Pondicherry. Sir has completed his MBBS from JJMC Davangere and his MD Physiology from KIMS Hubli. Sir has a teaching experience of 12 years in undergraduation and 11 years in post-graduation. His field of interest includes medical education, teaching, research, understanding the functions of human mind, and Sir has received the Nobel Medical Researcher of the year 2018 by MedWorld Asia International Publications for contribution in basic science research. I welcome you, sir, for this session. And I now request the chairpersons to introduce our speaker of the session, Dr. Chaya Gopalan. Over to you, sir. Thank you.
Yeah. Thank you, Dr. Vijaya Lakshmi. It's my pleasure to introduce the chairperson today, Dr. Chaya Gopalan, who is now the professor at Southern Illinois University, Edwardsville. She received her bachelor's and master's degree from Bangalore University, India, and a PhD from the University of Glasgow, Scotland, for a research on opioid peptides in the regulation of the release of luteinizing hormone. As a postdoctoral research fellow at Michigan State University, she continued her work on galanin and other neuropeptides. Besides teaching physiology at graduate and undergraduate levels for health professional programs, Dr. Gopalan has continued her lab research in neuroendocrine physiology. Currently, she is working on 2 research projects, one on the role of gonadal steroid hormones in the sexual dimorphism of the brain, and it is on this topic she will be giving her talk today. And the other project she is working on is on obesity, intermittent fasting, physical and mental exhaustion. Dr. Gopalan has received many awards, including Arthur C. Guyton Educator of the Year Award from the American Physiological Society, the Outstanding 2-Year College Teaching Award by the National Association of Biology Teachers, Excellence in Undergraduate Education Award by SAUC, APS Teaching Career Enhancement Award. She is visiting India as a Fulbright scholar during her sabbaticals to share innovative teaching methods such as flipped class has been my topic of interest too, where the lectures are shifted from classrooms as lecture videos, readings and practice programs, and the class time is used for discussion, group activity, and for formative assessment. Recipients of Fulbright grants are selected based on academic or professional achievements and demonstrated leadership in their field. Besides teaching and research, Dr. Gopalan enjoys mentoring her student and her peers. She regularly conducts workshops and participates in panel discussion related to higher education. Dr. Gopalan is very active in the teaching session of the APS and has served on many committees. She has published numerous manuscripts and case studies and contributed several textbook chapters and question banks for textbooks and board examinations. Over to you, madam. Eagerly waiting for your session.
This is a long and a really thorough introduction that was not required. Thank you very much, Dr. Janet Balin-Raj. Very nice to meet you virtually. Yeah, I would like to begin by saying my research interests have expanded from being focused on basic science only to exercise and nutrition and obesity, that's metabolic changes in the body and how other things can help treat these conditions. And then my original basic science research happened to be the neuroendocrine reproductive research, brain, how it regulates reproductive function. That's where I started with opioid peptides and then expanded to neuropeptides. Initially it was LH and then prolactin. And now, in addition to the basic science and other types of research, I am also interested in educational research. Flip teaching is one. If anyone wants any sort of introduction to flip teaching, please be in touch because I'm here and I'll be happy to talk to you. And the purpose of this trip through Fulbright is to introduce flip teaching in India. It's happening at one specific college, but I don't mind bringing it to a medical school like yours or at another group of people around you. So yes, if I could share my slides and share one particular area of research, I will go ahead and start. It's a very fascinating project. It's not anywhere near, taking it to humans or anything. It's just a thought of how one hormone can be so dominant, so dominant in determining the brain to be that of a male or to be that of a female. How any small deviation in the release of this hormone or the effect of this hormone at the target cell because of the lack of a receptor, because of the lack of an enzyme that prevents its conversion to a different hormone, so many things can happen behaviorally. Today we call these behaviors with different names, right? And that's where my research, this topic of research is also a very sensitive one. Very sensitive one, how a male physically can behave like a female, how a female physically can behave like a male. So it has a lot of potential, but there are lots of red tapes. I want you to keep that in mind and how it can be a difficult topic for me to secure funds or not secure funds. Depends on who is reading it and how I, take this sensitive matter to be a very, very serious matter, is where I am still pondering. Maybe you can help me, figure it all out. So let me get started with this introductory slide. I am in SIU Edwardsville, a huge campus, a beautiful, beautiful campus. I cannot say how beautiful this is. Very green, very peaceful. I spend some of my time outside, around the pond, meeting my students there, or faculty there, or that kind of thing. And I live in St. Louis, Missouri. If you think about the United States of America, that is where St. Louis or Missouri State is right in the center of the country. So we belong to Midwest. St. Louis is not far from Edwardsville. We are separated by Mississippi River, the long, long river that runs from the north to the south all the way. So I cross the river every day. It's just a beautiful drive, 30 minutes, 35 minutes drive to Edwardsville. Edwardsville is a university town. My city is like full of life, everything you expect in a typical city. And we are also referred to as the gateway to the West. West never existed for a long time. Everything happened in the East, Philadelphia, New York, DC, Georgia, etc. So now West started looking interesting. People did the gold rush, etc., etc. Anyway, the arch that you see here in the blue illustration is St. Louis Arch. That is the symbol of the beginning of the West, gateway to the West. So that's a little bit of introduction to my location. Now coming to this hormone, a magical hormone. So think about genetics. You know about XX, meaning female, XY means male. That is all set. You can't change any of that. So with XY, we see one thing. Okay, so interestingly, one thing you also have to remember is if there was no testosterone, the brain differentiates to become a female brain by default. Okay, so that is a natural phenomenon. And who changes it all? With the XY chromosome, you have the appropriate duct system that differentiates into testes. Testes at a particular time in a development would start releasing testosterone. Okay, so this testosterone is like the key to go and set the brain to be that of a male. All right, so how that happens and what can be done to manipulate this setup and what do we see when we manipulate is what I'm going to share. So testosterone is required in the setting of the brain to be that of a male. So that is in the initial phase. That's what we call as organizational phase. And then testosterone is needed again in the second phase, a continuous state that is beginning from the time of puberty. Around age 13, 14, 15 is when testosterone levels begin to climb again. So the initial kick, initial peak that you see is needed to set the brain, and then it disappears, low levels of testosterone until the age of puberty. And then from the time of puberty, testosterone is required to maintain that behavioral status. That's what we call as activational state. All right. So among many, many things testosterone does to the body, it does what we call as masculinization, male typical behavior. Okay. So how does it do that is where it also does defeminization, that is to remove the female-like behaviors and strengthen the opposite type of behavior. That's what we call as masculinization. All right. When the absence of testosterone, of course, is feminization. Testosterone has nothing to do with it, but I just wanted to introduce the opposite term just to be fair. And then, as I mentioned, any disruption in the availability of testosterone during the critical periods that I mentioned, that is organizational phase and the activational phase, could interfere with adult sexual behavior. It could more be like a female or certain grade of female, certain grade of male. So it's not just black and white. It's like a scale. How can you slide this scale to the right, which can be the male, or to the left, or in between? Those are all different versions of the male-female behaviors. So in the rats, where I have studied this extensively, the testosterone levels increase 4 days before birth and 4 days after birth. So that's what we call as the perinatal window, perinatal period, the critical period where testosterone must be present. Okay, in humans, you can equate this in a 40-week gestation period. So you may have a longer window of time where testosterone is needed right before birth. It could be a few weeks before birth, a few weeks after birth. Don't quote me on those dates because I do not study this in humans. But so you can imagine how a pregnant woman carrying a male fetus, releasing a certain amount of her body, her fetus is releasing certain amount of testosterone. So these levels are measured to confirm that this is actually true. So any disturbance in this release can interfere with the normal brain differentiation, is how you want to interpret that. So yet another interesting fact now, testosterone, although is the key hormone, he doesn't do the work. His work is done by his assistants. Testosterone can be converted to 2 different hormones. In the presence of 5-alpha reductase, testosterone becomes DHT, dihydrotestosterone. He is his assistant, one of his assistants. So his message is carried out by DHT, dihydrotestosterone. Or even more interestingly for you, testosterone can be converted to estrogen. I know some of you may be like thinking this is unbelievable. Can testosterone become estrogen? Yes, aromatase is the enzyme that converts testosterone to estrogen. So the perinatal testosterone that we looked at is now in the form of DHT or estrogen. And that's how it talks to the target cells. That's how it sets the brain into that of a male brain. So that is a fascinating fact. And believe it or not, this estrogen that reaches the cell may be one source, and for the brain to be differentiated into that of a male, there are papers that suggest high levels, toxic levels of estrogen is synthesized locally, such as in the hypothalamus, during these critical windows of time. That is the activational or the organizational phases. So this is all a factual detail that I'm sharing with you. This is not exactly my work yet. There are receptors that respond to DHT and estrogen. DHT is through androgen receptor, and estrogen hormone brings its effects through ER alpha, estrogen receptor beta. estrogen receptor alpha is supposed to be more involved in the masculinization effect, and both the ER and AR levels increase during the perinatal window, again suggesting they're hungry for the signal, and that's how they're going to bring about the hormones effects. Now, research question I have here is, if testosterone mediates its effects through these two hormones, DHT and estrogen, what role do they play specifically? Okay, can we isolate one hormone and keep just the other hormone and still see the normal effect or not, is where I have played with two separate approaches. One is to block androgen receptor. Okay, there is a drug called flutamide that I have used. I would use this drug during perinatal window. I would inject the pregnant moms with flutamide. Okay, so then that would reach the fetus and that would block the AR. That means even though there is testosterone, it would not be effective. Okay, so because now you're only blocking androgen receptor, you're keeping estrogen receptors alive. That means estrogen effects can go on as normal. Only the androgen receptors are blocked. Get it? So that's one approach I have used. And then in the second half, I have kept dihydrotestosterone active. I have not disturbed that, but then I have blocked the enzyme aromatase by using a drug, aromacin, that is XMS10. So this is how I have tested two separate drugs for two different hormones and wanted to see their effects. Behaviorally, in an adult rat. So this manipulation happened during the perinatal window, and then the behavioral tests were conducted later on. That is, as they became adults. Okay, so I mentioned that here. Organizational phase is when the manipulation is done. Adult rat behavior is what is studied. There are three different behavioral tests I have done: spatial working memory, open field, and sexual motivation. If there is a behavioral scientist here, they know what they mean, but if not, I will be explaining each and every one of these very soon. Okay, so the treatment I already mentioned. DAMs are the pregnant rats with flutamide or aromacin are the control groups received vehicle. Then behavioral, this is again, I have spelled out the dates. Perinatal means before delivery and the first few days after delivery. Okay, postnatal day 1 through 4 and prenatal is 18 through 21 days. And then we get timed pregnant rats, luckily. So we don't have to determine their dates. The labs would do that. So they're so accurate that I have never seen one that missed the dates. Behavioral testing is in a young adult, and that continues for a few months until all the behavioral tests are done. Postnatal day is 65 to 103. Sample collection is where we collect blood sample. I also collected the brain samples where I extracted hypothalamus, hippocampus, and amygdala. They are processed, semi-processed, and I still have to process some of those for receptor proteins, which I'll talk to you later. And from the blood sample, I measured testosterone. So that's the study plan. And the number of pups, you can't guarantee. Among the control animals, I had 10 male, 21 females. So that's where it was weird, more females than male. Aromacin group is where it was more or less equally balanced. Flutamide is where the numbers were on the low side. Again, we have zero control over the number of pups they give. birth to and the number of males and females they give birth to. So whatever I have to work with is how things go here. So I have tests that I would like to explain. You would place the male rat with a estrus rat and a ovariectomized rat. You can tell ovariectomized rat has low hormone levels. He's not going to be interested in the one with low hormones. Estrus rat is where in rats, the 4-day cycle is where estrus is where you have the peak of estrogen, a peak of LH, peak of FSH, and peak of estrogen. So that is when she is very ready for mating. So that is the period where I would pick and then place the male rat and see its behavior, if it is affected by the treatments I did or not. So that's that. Spatial working memory is a very elaborate one. You see 8-armed maze here, and we put food in each maze initially, and then we remove food from some mazes, but then the rat has to get used to knowing which arm has food, which arm doesn't have food. I have that shown here. A number of pellets. Habituation period is where all 8 arms would have food for 3 days. And then training is where only certain arms would have food, and testing is where we only have 1 place with food, not 3 places with food. So they really have to hunt for that food. The way they get used to is by going through this maze a number of days, and then now they have figured out. Where we place the maze, we have some visual cues. Apparently, rats connect the location to, oh, this wall where you have the color or the design, that's where I should go to get my food. Oh, this wall has no food, don't go there, it's that blank food. So that kind of visual clue is provided so that the rats know their ways through the maze. So now I'm going to talk about the results. One other behavior I didn't show here is the exploratory behavior. That is where you keep an empty space. You call that the arena. So it's like your cricket field or whatever with the borders, and then you place the rat in it. And if it sits in the corners, doesn't move a lot, that's the periphery, okay? And if it's a curious one, that's when you call it as exploratory. So that is when you know the animal is active, curious, normal. If it's sitting in an area, just not moving much, that means this rat is not exploratory, maybe depressed, etc. If it is too exploratory, this can be interpreted as anxious also. So the behavioral scientists have figured out how to use this technique, and I use open field is what we call the technique, and that's what I use. Open field testing is what you see here. We want to look at flutamide. Flutamide is the one that blocks androgen receptor, remember? So that is where you have flutamide is the gray one here. You can see what happened in the male. They were very curious, very exploratory, significantly different than any group studied. Whereas romacin, that is the one that prevents estrogen from being formed. There is no estrogen in their bodies, right? So they're not as curious. That is basically what you're saying. So estrogen is linked to exploration. So I think you have a way of saying that maybe when you block the testosterone from being converted to DHT, or when androgen receptor is blocked, more of the testosterone could be converted to estrogen, because there is estrogen, more estrogen, right? And so that is now being used in the exploration is one explanation I can come up with. So sexual motivation is where it's very interesting. Okay, time spent with OVX. OVX is ovariectomized rat. You can see how the control and the treatment groups, nobody really likes the OVX group here. And all male rats run to estrus. Estrus rat. It's basically experiment is working. Estrus rat is hot, and they all want the hot rat. And then here, aromacin group is where, remember what's happening, there is no estrogen. It's mainly DHT. So more DHT means more male-like masculinization type of behavior is what we noticed. So that is very interesting. So both the exploration and sexual motivation had some interesting data. On the other hand, spatial working, both groups, all groups behave the same. Females were more, had a trend towards having better memory of the location of food. Males were slacking is what we noticed, but it's not significantly different, so I can't really prove it. This is hormone level in adult. So this is interesting. What happens to testosterone in an adult with your manipulation during development or perinatal period? The one that received aromacin, that is where estrogen is not available, there is more testosterone. It continued to make more testosterone. It looked like we don't have the points in time, but it's only one point. That's when the animals were sacrificed. We were able to pick up the levels. So this is GCMS technique we used to measure testosterone. Aromacin increases testosterone. So in my conclusion, okay, I already gave all the key results. Now, dihydrotestosterone and testosterone, when there is no estrogen, okay, that is only in the perinatal period. We have not worked on the activation period. Appears to play a role in enhancing socio-sexual behavior during adulthood. Okay, socio-sexual behavior. So that is the increased interest in a female. All right. Male rats receiving flutamide, that is where you do not have the androgen receptor to signal the testosterone or DHT's message to the cells. So mainly the estrogen signal is possible. They spent a longer time in the middle zone, meaning more active state of the animal compared to the other groups. Okay, so estrogen mediated effect in early stages of development appears to last through adulthood. That is an interesting observation. Estrogen is associated with increased exploratory behavior. All right, blocking AR during perinatal period elevated exploratory behavior in adult male rats, again suggesting there is estrogen effect, and absence of AR-mediated effect during early part of life appears to allow estrogen to be major hormone regulating exploratory behavior. We talked about that. Estrogen deprivation would increase testosterone levels in adults is another point I'm making here. So the next step is, more or less this is done. Flutamide work is done. Aromacin brains are processed. I still have to do RT-PCR to do the receptor protein expression to see how ER or AR expression altered in these groups to be able to connect what we saw behaviorally with the molecular changes that we see within the brain. I have chosen 3 areas: hypothalamus, amygdala, and hippocampus. Hypothalamus is the most central part in the regulation of sexual dimorphism. Hippocampus is more for the memory, since we didn't see much memory effect through spatial working memory test. I'm not expecting a whole lot, but we will see. Amygdala is probably a neutral one here. It's more of an emotional center. So we are not sure what to expect in there. If anyone wants to collaborate on any of this stuff, you're more than welcome to contact me. I'll be happy to do my level best to involve you here. All right. Where do you go from here is probably what you're going to ask me. I'm going to suggest a few things, and any ideas are welcome. One, I would like to repeat the whole study of manipulating DHT and estrogen at the activational state, that is puberty onwards. I would pinpoint a young adult stage and see how the behavioral changes would be seen in this manipulation. There are studies done previously, in 80s and 90s actually, but I would like to do this one round and then add molecular component to this. And I would like to study the knockout of both ER and AR. Testosterone is released, but no ER, no AR. What would this animal do in that situation is another interesting thing. I mean, I'm hoping to do. For nanomolecular biologists, if there are any here, I have suggested tagging a very fine particle to testosterone as it is released. You're talking about fetal rat, so tiny, 1 gram in weight, for example. I don't know how you can do any surgery tagging in testosterone. That's the tricky part. If that could be done, we can trace this testosterone and see where exactly it goes, how exactly it changes, is going to solve a huge puzzle. Okay, so this is giving away my idea to other nanobiologists and other reproductive neuroendocrine physiologists here to take it on and solve the puzzle. All right, so I would like to hear what you have to say. Before I conclude, I would like to thank a number of people, my students, who have worked hard to do some of the animal work. It is very time consuming behavioral work. I do it, but I also like to teach students, and they get excited and they want to do more and more. And all the discussion that we have is just phenomenal. And I would like to thank Rupadai. We do Medical College and Hospital, Puducherry, for providing this opportunity to speak to you today, and in particular, Dr. Lakshmi Chatia, who was so instrumental in tracking me down, whether I respond to her email or not, and make sure that I'm here today doing my job. And I sincerely thank you all for listening to me, and I am open to questions. Now I'll take the slides. You did already. Okay.
Okay, Dr. Janet. Yeah, I see that you have some questions.
Yes. Thank you, madam, for highlighting us on how testosterone functions at the perinatal age, how testosterone is converted into DPH and E2, and how best we can knock out the enzyme aromatase using aromacin and flutamide, so that we can change the morphism and then check how best the animal will decide. But since a person who have been working on behavioral changes in pre-diabetes, I'm a little bit concerned with I have few questions for you. Why was spatial memory and open field arena, social behavior, exploratory behavior checked in these animals which you tested? Because usually what happens is using open field arena, we usually don't find much of difference between male and female animals. But in study, it was very obvious that male were more exploratory when compared to females. So is there any reason behind it? Because we see it as nocturnal animal, not like any other. Yeah.
Why we tested open field exploratory behavior? We were wanting to make sure that, you know, testosterone male animals are typically exploratory, and do we keep that behavior going or not? No, you know, what if they are going to be shy and sitting in one place, not moving as much? So that was one of the reasons to do that. Spatial working memory is directly linked to estrogen. Okay, when we turn off the HT's effect, is there more estrogen that is potentially around, and would that emphasize, you know, better memory so they can track down the food better, you know, that kind of a reason.
The future researches are very promising.
Oh,
it will be very interesting.
Is there any volunteer to come up and collaborate? And if you know somebody who is interested in this field, you can share my contact and we can talk about it. I'm here and I can guide in whatever capacity I can. If anybody is coming to the States or wanting to come to the States, I can, you know, guide them to see how they can pursue that.
That
Yeah.
was
Thank
really
you.
nice, ma'am. Yeah, ma'am, maybe by next year when we are organizing our next conference, probably you'll be ready with your results.
At the receptors.
Yeah.
Yes.
Anyone else has any question? The next speaker is already in. So we would now, it's time to honor our guest speaker for today, Dr. Chaya Gopalan. It was very nice to hear you, ma'am.
Thank you
Really
so
wonderful.
much.
Yeah, thank you.
Thank you.
Unmute, ma'am, please.
Yeah, pardon me. Dr. Tilly, you have a question?
Ma'am, he's currently not available, ma'am.
Oh, he has left. Okay, fine. Okay. Next, we would like to honor our chairperson, Dr. Janet Barin Raj. And Dr. Shiv Yogapa Telly. Thank you, Dr. Chaya Gopalan, for this wonderful session. Over to the next session. Now we have next session from the Department of Biochemistry. Okay. Over to the moderators for the next session.
Thank you.
Thank you all.